Hi, everyone. My name is Lisa Mesrop, and I am a third-year graduate student in Dr. Todd Oakley's lab at UCSB. And the title of my presentation today is Characterizing the Genetic and Molecular Basis of Bioluminescent Production and the Origin of the Light Organ Bioluminescent Ostracods. And I am a Wrigley Fellow. I was given the award last year, but I wasn't able to go out there because of COVID. And so finally, thankfully this year I was able to go out to the island. But yeah, so I'm going to start my presentation. Let's see. And so here's an introduction. So I'm broadly interested in how organismal complexity originates. So complexity can be defined as a system that has many different parts that can translate to any level of biological organization, such as cell types, proteins, and genes. And a complex system like bioluminescent glands are made up of many different parts. So they have many different types of cells, different types of genes and proteins. And my interest lies in understanding how did these parts, such as these cell types or new cell types, how did these parts originate and evolve? And so there are some representatives here from different taxa that have these specialized secretory glands, such as the salt glands in snakes, the defense gland, rove beetles, and bioluminescent glands in ostracods. These are specialized secretory glands, and they're kind of like a hotspot for studying novelty. So you can think of these glands as having similar secretory mechanisms as a function of secreting, but they have lineage-specific proteins and small molecules that have originated within that lineage, and they can use it for defense. So my two broad questions are, what are the molecular mechanisms underlying the evolution of novel glandular systems, and how are novel secretory cell types generated and assembled into organ-level structures during development and evolution? And so my system that I'm interested in asking this question is in bioluminescent ostracods. So I'm interested in understanding this novel light organ. All bioluminescent ostracods have this light organ that produces the light emitting compounds. And I'm interested in understanding, how do the cell types that make these light emitting compounds, where do they come from? How do they originate, and how do they come together during evolution to produce a organ level structure? And so here on the picture, this is a bioluminescent ostracod. They're really, really tiny. It looks pretty big in this picture, but they're super tiny. They're the size of a sesame seed. And illustrated here by the circle is this light organ. And a really cool paper done by Hugard back in 1990 did this really cool histological study trying to characterize the cell types within this light organ. And the study had found that there are two specialized cell types, one for producing the substrate, luciferin, and the other cell type specialized for producing luciferase, which is the enzyme. So both the substrate and the enzyme is needed for the bioluminescent biochemical reaction that occurs extracellularly. So ostracods secrete these compounds in a mucus-like substance, and then the light reaction occurs outside of the body in the surrounding water. And it's really, really bright and blue. And so how does the bioluminescent biochemistry work? Well, so in the previous slide, you have these two specialized cell types that produce the protein and the substrate that make up the light organ. And the protein is called luciferase, and the substrate is called luciferin. And when they combine, you get this beautiful bioluminescence. And this video clip here actually are the courtship signals of bioluminescent ostracods that use bioluminescence for courtship signaling, which is really, really cool. And so different species will have different patterns of this bioluminescent glow, and it's beautiful when you get to see them underwater. So one really cool, one important thing though is the biosynthesis pathway that makes up the luciferin bargulin has not been characterized in any bioluminescent animal except for bacterial and fungal luciferin. And sometimes I also I love to show this video because I think it really is a cool video. I'll replay it again, but it is a very cool video demonstrating one of the functions of bioluminescence. And so here you have an ostracod and you have a cardinal fish, and the cardinal fish attempts to bite the luminous ostracod, but then it spits it out. And you'll kind of see that the ostracod gets into the mouth of the cardinal fish, and then it secretes the bioluminescent glow or mucus, and then the fish spits it out. So here's the ostracod, and the fish swallows it, but the ostracod is spewing out bioluminescent mucus, and then finally it spits it out, and the ostracod makes an escape. So it's pretty bright, but it's really cool. All right, so from a recent from a recent phylogeny, we know that there is a single point of origin of bioluminescence within the Cypripedidae family. And the tips represent non-bioluminescent, the white tips represent non-bioluminescent ostracods, and then the blue tips represent bioluminescent ostracods. And the yellow point here represents a single point of origin. And so the pink circle here actually represents the ostracods that are found at Catalina and the ones that I study, and also non-luminescent ostracods are also found in Catalina as well. So all ostracods have this homologous upper lip gland. So the light organ is actually within this glandular kind of muscle, which is called the upper lip. And all ostracods have it, non-luminescent and luminescent ostracods, but only luminescent ostracods have the light organ within the upper lip gland. And so this allows us to understand the changes in gene expression that generates novel specialized cell types at this transition here, at this origin point here, from non-bioluminescent to bioluminescent ostracods, and kind of help us to deconstruct the evolution of the light organ gland. And so why Wrigley? Well, the bioluminescent ostracod Rhygulosugii is the only bioluminescent species found in the Pacific coast of North America, and the Wrigley Pier is the only location known to consistently have high populations. So it's pretty cool. It's pretty awesome that I get to go out there with the Wrigley Fellowship and collect as many as I can get. And so my research objectives, or my summer plan at the Wrigley Institute, involves the collection of bioluminescent ostracods to address my research question. And what I'm really interested in is understanding the molecular and genetic basis of bioluminescent mucus production and the origin of the light organ in ostracods. So our lab has identified and functionally tested the luciferase gene, but the biosynthesis pathway that makes the luciferin molecule is unknown. And so I hypothesize that genes involved in the luciferin biosynthesis pathway and the production of secreted mucus are actually co-regulated with luciferase. So in my project, I used a large-scale gene co-expression profiling to identify gene candidates that could potentially be responsible for the production of luciferin molecule and also responsible for the secreted mucus. And so we would expect if genes comprising luciferin pathway and luciferase are co-regulated, then the genes in luciferin pathway, then we would expect the genes within the luciferin pathway to form tight associations with luciferase genes in co-expression networks, facilitating their identification. And so that was my research question, my broad research question, but in order to test or in order to explore that research question, I need to collect a lot of bioluminescent ostracods. And so how do we do that? And so we collect them in these PVC traps. So we have these PVC traps, and then on both ends, we put like a mesh, and we also put like a little mesh funnel, allowing ostracods to get into the PVC trap, but not get out or not escape. And we usually bait the traps with chicken liver, and the traps are set in about probably 8 to 10 meters of water right after sunset, and they're collected roughly 2 hours after sunset to allow ostracods to accumulate. So when I was out at Wrigley, I would set up all my traps, and then I would go down to the pier, and I'd watch the sunset, wait till the sun sets, and then I would set out my traps, and then I would come back 2 hours later and I would retrieve the traps. I'd go up to the lab and then I'd start processing the traps and start separating the bioluminescent ostracods from some of the other little critters that would also get into the traps. And so with all the animals I had collected, my project, I performed weighted gene co-expression network analysis. So I took expression profiles of a lot of different bioluminescent ostracods, or Vargulus sugii specifically, and I subjected them, not me, I also work with 2 fabulous undergrads and a postdoc, and we retrieved expression profiles or transcriptomic expression profiles from ostracods at various stages during the day and at night, and also of the upper lip gland and whole body, and also other non-homologous organs. And then I had performed this weighted gene co-expression network to understand some of the genes that are being co-expressed with luciferase, to understand the biosynthesis pathway of luciferin, and also learn a little bit more about the other genes that are being co-expressed with luciferase. And so the way that we did this is we retrieved probably about like 50 expression profiles from males, females, and ostracods under different different conditions, and then we obtained all their gene expression values. So first we start with a matrix that has all the samples and all the expression values, and then from there built a similarity co-expression network, and then the modules or groups of genes that are highly co-expressed are then generated by using hierarchical clustering, which groups genes based on similar expression patterns in a pairwise fashion. So here on the right, the dendrogram is showing this relationship of all the genes. The height represents similarity, and the modules are assigned by cutting the tree and specifying height, and branches below that cut height become modules of highly co-regulated genes or co-expressed genes. So each of these different colors here are presumably different modules of genes or highly co-expressed genes that may function together or have some sort of biological relevance. And then from here, then you can build a network, and the nodes here represent genes and the edges represent their similarity score, or how the weight of how correlated these 2 genes are based on the analysis. All right, so we did this. We took all the expression profiles, we built a gene network analysis, and the bioluminescent network here is a subset of the entire network, but it represents a group of co-expressed genes that are associated with the production of bioluminescence in the upper lip gland. And the Vargulus sugii luciferase gene is shown here in the middle, and the edges are represented by the gray lines and include the genes that have a direct relationship with luciferase. And genes that are co-expressed with luciferase include genes that have BWD domains, which is a characteristic of mucin proteins or digestive proteins, and a significant portion of genes that are related to venom and toxin-like genes, and genes that are also involved in protein folding and modification, such as genes in the UPR or ERAD pathways, which are kind of like quality control pathways involved in protein synthesis. And so, secreted genes such as the venom allergen, which are known to elicit inflammatory response, and genes that have this disintegrin and metalloproteinase motifs, which are part of the metalloproteinase family, are genes found in venom cocktails secreted by other venomous animals. So both of these genes are highly upregulated in the upper lip gland compared to the eyes and gut, implying that these genes are of greater functional relevance in the upper lip gland. So we found a lot of these highly co-expressed genes with luciferase to also be upregulated in the upper lip gland compared to non-homologous gut and eye gland. And that's pretty cool because it's kind of suggests that maybe the bioluminescent network may have been co-opted from this conserved venom network that exists in closely related crustaceans or venomous crustaceans. So that's a little bit of my preliminary work, and I think I'm a little over on time, but I just wanted to thank everybody and thank you, Wrigley, for your contribution, allowing me to perform my research at USC Wrigley Institute. Thank you to my fabulous lab, my advisor, Dr. Todd Oakley, and postdoc Jessica, and the two undergrads that I had worked with, Michael and also Gigi, and just want to thank everybody, and grateful to do this awesome, cool science and be part of a great lab and have the opportunity to go out to Wrigley to collect ostracods.